Okay, so we are having a discussion section now, so please ask questions. Especially we encourage the young scientists here to ask questions.
So, you can hear me. So, thank you for a wonderful session. Now, when I see Michele and Ashok to sit there side by side, so both with a very, very long experience, one with gene therapy and one with cell therapy. So I would like to ask that you both answer to my 3 questions. So first I would like you both to grade from 0 to 10, both the gene therapy and cell therapy, how close they are to clinic. 10 is very close, 0 is really far away. My second question to both of you is, which one you think will enter the clinic first and when? And my third question is, I mean, nobody of you really mentioned anything about the adverse events of the gene therapy or cell therapy. So I would like to, somehow address this question. What you think, what might be the adverse events that one had to wait relative to possible beneficial effects?
Should I go first?
Okay. 0 to 10. I should vote for cell therapy also. Both.
Yeah, both. So I would like you both answer what you think about gene therapy and cell therapy.
Okay. So I cannot give really high scores to either one of these. I would give a higher score to gene therapy, though. You want numbers? 7 and 6, I don't know. I think the first one that will get into the clinics, if I had to make a bet today.
Is give also the basis why you think that you give higher score for gene therapy than cell therapy?
Well, the reason I think that gene therapy may get into the clinics first is that, and actually this is a question that I wanted to ask to Ashok, is to my knowledge, right now, cell therapies produces essentially bystander effects, right? I do not know any solid evidence of integration in circuits and repair of circuits based on cell therapy. And I was wondering actually if Ashok believes that these data are based on integration in the circuit or bystander effects.
Yeah, I mean, bystander effects are there. You see many bystander effects, but they're all beneficial. But some studies have also shown integration of transplant-derived cells. For example, with the transplantation of GABAergic neurons done by Scott Baraban, recently published in Nature Neuroscience. And they do show the integration of GABAergic neurons into hippocampal circuitry using electrophysiology techniques. So whatever new GABAergic neurons they are adding into the circuitry, they get hooked up with the hippocampal principal cells. But there is one mechanism for suppressing seizures. But the neural stem cells, they can do much more than that. So they can secrete a variety of factors. For example, GDNF is an anticonvulsant factor, and which is greatly downregulated in chronic epileptic conditions. So you can bring that level back.
So yeah, I agree that the paper by Scott is the first
Yes.
that provides some strong evidence of integration. What I'm not sure about is that you get a sufficiently widespread integration of cells in the circuit, even in that case.
It depends on where do you transplant, because they don't migrate a lot. But in Scott's paper, I think they tried to place transplants into the dentate gyrus so that they can hook up with the granule cells, to decrease hyperexcitability there.
Yeah. But in terms of which will go to the clinic first, if I have to bet today, I would bet on MPY vectors in pharmacoresistant patients. And the reason I'm saying that is that there is a robust anti-seizure effect, that it is ethically feasible to inject at least adeno-associated vectors in part of the brain that eventually can be removed, and that MPY can produce robust effects also in human slices, which is at variance with galanin that is not robust, apparently.
So if I can jump in with a point for the cell replacement transplant therapies, I guess one issue thinking long-term is that particularly in patients who've had epilepsy for a long time, a lot of these cell populations are dead. And that I think the potential of being able to restore some of those populations may give you additional benefits beyond controlling seizures, where I think some of the gene therapy approaches are trying to either decrease excitation or increase inhibition.
Yeah, but here comes the other problem, and that probably...
I'm not saying it's easy.
No. That is the environment. The microenvironment there is clearly a microenvironment that does not favor survival of the cells, because cells are dying. You have
I think the hurdle is huge, but ultimately that's kind of what you'd want to do, is to restore the circuit.
Yeah.
And that's
what Ashok showed is that he gets survival of a huge percentage of his graft. I'm sorry, is very promising.
Yeah, but gene therapy also has problems. My question is, when you infect these cells with all these viruses, how long they last? I mean, is that a transient effect or a permanent effect? That's the biggest issue. Yeah.
Well, in the case of herpes, the effect is transient, and that is probably dependent on the fact that the virus does not insert its genome in the host cells, which is good because you avoid the risk of insertional mutagenesis, but at the same time, the episome gets silenced spontaneously, and you don't get a long-lasting effect, which could be an advantage in the case, for example, of trophic factors, as I showed, but it's obviously a hurdle for other kind of applications.
Now, if you want to
adeno-associated lentiviruses can last for years.
Last for years. Yeah. So what are the side effects of this kind of therapy?
The evidence with adeno-associated vectors is that there is no major side effect.
Sorry to interfere, but I mean the issue is what has been actually has that actually been clearly addressed in these studies? Being honest with you, I don't think that this issue of adverse events has been vigorously addressed.
No. I agree. And I think that both gene therapy and cell therapy are still having that problem.
Okay, so you say gene therapy 7, cell therapy 6, gene therapy goes first, and adverse events should be investigated better. So Ashok, what are your numbers?
Yeah, I'm going to give 6 for both, because both have problems. Yeah, so I'm not going to take any sides here, because gene therapy has problems. And based on the studies conducted so far, the amount of suppression of seizures you get is not really that dramatic. So is that enough to treat a chronically epileptic patient? That's my question with gene therapy. And cell therapy is a developing field. There are issues, particularly what kind of donor cells we need for transplantation in patients. Ultimately we have to fall back on embryonic stem cells or induced pluripotent stem cells, that has a potential for doing patient-specific stem cell transplantation. You can just take a skin biopsy from a temporal lobe epilepsy patient and reprogram skin fibroblasts into pluripotent stem cells and then push them into any lineage you want, either GABAergic progenitors or neural stem cells, and you can transplant that. But there are problems, because in your cell suspension, even if you include one pluripotent stem cell, there's a potential for developing a teratoma. So you don't want to have teratomas, after transplantation. So there are problems, but the iPSC field is really progressing now, and then people are coming up with really novel ideas, to isolate, the cells they want and to avoid pluripotent stem cells in the suspension. There are some ideas about, inserting suicide genes so that if they start proliferating after transplantation, you can kill those cells, by activating these suicide genes. But the field is, moving with great caution. But there is promise.
So what about the adverse events?
Accuracy. What is it? Accuracy.
Accuracy.
Yeah.
Accuracy of what?
Accuracy of the treatment.
Accuracy of the treatment?
Yeah.
No, what kind of side effects you can imagine? Because I don't think that...
The side effects...
Because I was watching very carefully the presentations, and like in papers, people like to present only the positive, favorable data.
Yeah.
But people don't even really, including, myself, we don't really do vigorous adverse event
Now, what kind of adverse events you're expecting? Because based on whatever we have done so far, we see mostly beneficial effects with neural stem cells. They can do a lot of things. They can suppress abnormal neurogenesis. They can induce neuroprotection, and and some other studies have shown that they can also suppress inflammation. We haven't examined that.
Yes, but I'm just asking, do you actually do regular monitoring of those animals to watch if they actually develop some adverse events? Because if one doesn't look, one doesn't see it.
Yeah, based on the behavioral tests we have done so far, we haven't seen any adverse effects. Whether it is cognitive function tests, depression test, we always found beneficial effects of this transplantation. And in terms of seizure suppression also, it's always beneficial effects. We haven't seen any, increase in seizures, for example, after transplantation. That is one of the things people who do not do transplantation, they always ask me, transplantation might cause seizures. That may be true with certain cell types, but with the cell types we are using, we haven't seen any increase in seizures.
Yeah, the same for me.
Actually, I have a question. I think maybe you just answer now. Have you looked at the process inflammatory in the hippocampus after the implantation of this, stem cells? Because we know that maybe they can suppress the inflammatory process. And another question is, the new cells, have you looked at the density of the spines? Because we know that after status, the density of the spines in the granular cells may change. And have you looked at this?
We haven't examined the density of spines, in chronic epileptic conditions or after transplantation. We haven't done that. That is a very interesting question. That's something to examine because a lot of plasticity occurs after status epilepticus, even in chronic epileptic conditions. So it's good to examine in the future. Yeah. What was the first question? I didn't get.
About the inflammatory process that we saw in the hippocampus after the status epilepticus. If the stem cells suppress this inflammatory process.
Identified the effects of neural stem cell transplants on inflammation, but there are other models like stroke models. Swedish group, they have shown very beautifully suppression of inflammation by neural stem cell grafts, actually by the very same subventricular zone neural stem cell grafts.
Ed has a question.
Question for McKelly, please. Is the thought that the viral DNA, once that's been inserted into the cell works completely independently of the host nuclear DNA and the host mitochondrial DNA, or is it possible that there's some transcription or protein expression that goes on between different sets of DNA?
I'm not sure I understood the question. So you're asking if the viral DNA could produce some unexpected effect depending on...
Is there any interaction between the inserted DNA and the function of the host DNA in any way?
That's in principle possible, to the point that they can induce neoplastic transformation of the cell, so induce the production of tumors. But it is a sort of a random effect. Depends on where specifically the DNA gets inserted, and there is no possibility to establish in advance where it will get. Did I answer questions?
Thank you, all the speakers, for the interesting lectures. And my question is similar to his questions, but I was thinking, Professor Ashok, if there is any evidence about mechanisms underlying, there is evidence of various different mechanisms underlying alterations in temporal lobe epilepsy in humans. How can we can be sure about transplanted cells not being suffering these effects in humans? I mean, if the process, the altered process, will going on after the transplant and maybe cause disturbance even with the transplanted cells?
Yeah, that's a good question. I mean, if transplanted cells, they continue to proliferate, they can fill up the hippocampus, that can be a problem. And most of the stem cells which are obtained from the postnatal or adult brain, they are committed cells. They, after transplantation, because of differentiation factors, they typically differentiate into either neurons or glia. And we don't see much residual proliferation in the transplant. So all of them differentiate. But if they continue to proliferate, that can be an issue. So that's why we are doing long-term studies after transplantation. So in this, in the second model, we actually examined the phenotype of stem cell-derived cells 9 months after transplantation. So we did not see any proliferating, the grafted cells. It did not proliferate. So they are all differentiated. But if you do embryonic stem cells or iPSC cells, that can be an issue. So that's why it's important to pre-differentiate these cells and then sort them and then transplant.
Comment and a question for Steve Downser. Steve, I wanted to first of all applaud your efforts in now trying to do these electrophysiological studies on these ectopic neurons. My question really relates to what do you think that these ectopic cells are referring to a more juvenile form? For example, there's been a lot of work recently where people have described the shift of GABA to a depolarizing form in human tissue, and the whole idea that GABA becomes excitatory. And also we know that juvenile neurons have a very different expression of NMDA receptors, which makes them more excitable. And then the second thing, which is maybe more of a comment with respect to these doublets, I mean, one possibility is that you've got an increase in low threshold T-type calcium currents potentially, but also there may be alterations in the electrogenesis in the somatodendritic compartment, which is potentially leading to amplification of inputs and causing these doublets to occur.
So on the first question, what he's referring to is that developing neurons have altered chloride gradients because of the chloride transporters, and can actually be excited by GABA. So we normally know GABA is an inhibitory neurotransmitter, but very young cells, it seems like they can be excited because of this reversed chloride gradient. So we're looking, these studies were done on mature cells, so we would presume that they have the mature chloride gradients, but we haven't done the experiments to test that out specifically, so it's certainly possible and maybe something interesting to look at. So on remind me on the second question. Oh, yeah, on the, yeah, so we haven't, again, we haven't done the experiments yet to really, so we've seen the phenomena, but we're going to have to do some experiments with different pharmacological agents to try and tease that out. And I agree that perhaps changes in the morphology or some of the ion channel types of these cells could account for that.
Hi, this question is about the cell and gene therapy again. And do you think that one of the barriers to the success of this, regardless of the technical considerations or just overall safety to the patient, is the fact that we would need to understand the genetics better first? Because as I see it, to do gene therapy, you'd have to know what gene is dysfunctioning. Or for cell therapy, if you want to do an inducible pluripotent stem cell, you can't just, it seems like you couldn't just implant someone's own cells in that already have the genetic code that's faulty that's causing the epilepsy in the first place. You would have to almost like do gene therapy on their own cell first before you could put it back in. So I just wonder if you could comment about that.
I was talking about patient-specific stem cells for temporal lobe epilepsy, which is not genetically related. But for genetic related epilepsy, I don't think we can do patient-specific stem cells.
Yeah, I also said that genetic epilepsies are are very unlikely to be a target for gene therapy soon at all. And so what is probably has a better chance to work is to induce production and secretion of soluble factors. So you're talking of therapeutic genes and not a real genetic intervention.
So I have a question to Michele. So why you chose BDNF? Because some of the earlier studies by Helen Schaffman showed that BDNF actually increases seizures. So, and also after status epilepticus, there is an upregulation of BDNF, almost every cell layer of hippocampus. Yeah. So why do you, think BDNF is suitable for gene therapy?
And Danzer works too. So Steve have published on BDNF too, showing this, right?
Yeah, that is something that, of course, is a problem. Well, first of all, we do not use BDNF alone. In vitro, I didn't show the data we had in vitro systems, but I showed that we produced also vectors expressing BDNF alone and FGF2 alone. And in vitro, neither the one with FGF alone nor the other with the BDNF alone exert significant effects, while the two together do much better. As for the time course of overexpression, we actually induce overexpression of BDNF when it's down. The initial burst is gone. And the question that remains open is one way or the other, BDNF is a good guy in my hands, and it is a bad guy not according to Ellen, but Jim McNamara also recently published a paper in Neuron on that part. So we have been thinking a lot about that, and there are multiple possible explanations. One is related to the receptors involved. And there is track B, that is the high affinity receptor, but is also P75, that is the low affinity receptor, that has been demonstrated to produce effects that are opposite. So, and it's mainly activated by pro-BDNF, so the processing could be a factor. But one hypothesis that I like, because it is related to my previous work on BDNF messenger RNA, is that there could be also a matter of splice variants. So BDNF mRNA can be delivered to dendrites in an activity-dependent manner, and that induces biosynthesis of BDNF near synaptic contacts. So it is likely, I think, that the negative effects of BDNF may be related to potentiation of excitatory synaptic contacts, while the positive effects could be related to trophic effects, right? So it could be that depending on the site of the neuron where you have a biosynthesis of BDNF, you can have an opposite effect. And this is consistent with the biology of BDNF, because if you look at the structure of the gene, you have an exon that contains all the coding sequence and 8 other exons that are alternatively spliced. And they are independently regulated by different promoters, meaning that they get activated by different inputs. And what we found is that some of these splice variants target BDNF to the dendrites, and others instead are, in spite of being activated by seizures or by status, both kinase and pirocarpine, produce BDNF only strictly in the cell body. So what we are trying to do now is to produce anti-sense products to, and those are vectors as a matter of fact, to specifically interfere with the splice variants. And it would be nice to see if we could dissect out these pro or anti-epileptic effects.
Sounds very good, yeah. So I have a question to Steve also. So you mentioned that in your P10 knockout mice, with 5% of newly born granule cells going into the hilus, you found seizures, right?
So 5% were P10 knockout, and then a small subset of that become ectopic. Okay. But the 5% was the number of P10 knockout cells of granule cells.
Granule cells,
yeah.
Okay. So with that, you found seizures in these animals. Mm-hmm. I mean, did you see any other abnormalities in the hippocampus?
So grossly, the hippocampus is normal. So these animals, the P10 knockout cells, as I showed, they're hypertrophic. They get basal dendrites. Some of them are ectopic. But in the animals with 5% knockout, we don't see mossy fiber sprouting. So it's another example of a dissociation that mossy fiber sprouting is not required, at least in this model, for epileptogenesis.
Did you see any cognitive problems in these animals?
We're looking at that now. We're not sure yet, so we're still waiting on the data.
So, if Simonato allows me, I would like to switch the score like 7 to 5 to cellular therapy, essentially because of the failures in the cystic fibrosis clinical trials. So this puts away clinical trials for gene therapy for many years, I think, in the future. So I would bet I will put like 7 to 5 to cell therapy. And since NPY looks like a good option, I would ask Dr. Shetty what if he thinks about putting cells that secrete NPY.
What is the question? Is it about gene therapy?
No, use cells to secrete NPY. Neuropeptide.
Cells secreting NPY?
Yeah.
Put those in instead.
Okay. So, I mean, if you transplant neural stem cells, only a small fraction of them will become NPY. But what we found was the host NPY-positive neurons, which otherwise die after status epilepticus, they are rescued with transplantation. Okay, that's why we have increased number of NPY-positive neurons in the hilus in transplanted animals. But it is possible to, genetically engineer neural stem cells to secrete NPY also. That is another option, yeah.
For Andy, in your experience in the clinical aspect, is it possible to reduce the seizure activity with drugs with the antidepressant effects?
Yes. So the question is, can we reduce the seizure frequency with antidepressant drugs? There are several open trials that have used SSRIs in patients with intractable epilepsy, where there's been a reduction in seizure frequency. But those were open trials. We, at our center in Chicago, completed a study and found a similar finding. I think that this is something that needs to be investigated, and we are, planning a grant, a multi-center study grant, where it needs to be tested against placebo in a double-blind placebo-controlled study against placebo. In order to make a case for that. In theory, I think the proof of concept exists. There is cellular data in the sliced preparation of hippocampus that shows that SSRIs decrease hyperexcitability. In animal models, there are multiple animal models that show that SSRIs block seizures, and this is not only in the rat, but also in rats, cats, including the chimpanzees. So there is proof of principle, but we need to test it under double-blind placebo-controlled trials.
But do you think that the international companies selling anti-epileptic drugs that are very expensive in some countries will support this idea?
One of the obstacles, because when, the only way you can do this study is by funding from federal agencies, because no pharmaceutical company is going to invest in doing a trial like this, because all these drugs are available in generic formulation. So the only way we can do it is if we can get funding from some kind of federal agency. What I can tell you, and this is anecdotal data, it's not double-blind control, but I pay close attention to patients with a history of depression and anxiety and their seizure disorder. And I have been impressed by the fact that when patients have a history of depression that we've treated with medication or not, and they have recurrence of seizures, often the recurrence of seizures follow the recurrence of depression. So first they get depressed again, then the seizures come back. So it's not that they get depressed because the seizures came back. First they get depressed, then the seizures come back. But again, this needs to be tested, prospectively, and other than that, it's anecdotal.
The question that is actually comparing the stories by Dr. Dancer and Dr. Shetty. So Dr. Dancer showed very elegantly that these ectopic cells form a risk, for instance, for seizures. And yet when I looked at the grafted cells in Dr. Shetty's presentation, these cells were in the weirdest places. They were not in where normally cells are in CA3 area. They were just beside the pyramidal cell layer. So I have a question to Dr. Shetty, what you think how these cells are integrated into the circuit so that they give all these beneficial effects, and to Dr. Dancer to comment on that. How is that possible and may it be different for CA3 compared to dentate? I'm just puzzled by these two results.
Yeah, so we examined neuronal differentiation of transplant-derived cells very carefully. So I mentioned that about 20% of cells, they migrate into the granule cell layer. And such cells, they differentiate into granule cells. They don't express any GABA. But the cells that migrate into CA3, CA1, hilus, they differentiate into neurons. About 80% of cells are there. Out of that, 25% of them differentiate into GABAergic neurons. So they don't make glutamatergic neurons in those areas. Yeah. So chances of them generating seizures is unlikely because they're going to hook up with the principal neurons and probably suppress seizures. Yeah.
I would agree with that interpretation that what we're seeing in our epilepsy models is these granule cells, which are glutamatergic excitatory neurons migrating to the hilus, and they are forming the wrong kinds of connections, and Vic Nadler has done some elegant work recording from some of these ectopic granule cells, and they have abnormal firing patterns and properties consistent with them promoting hyperexcitability, whereas Ashok cells are, as he said, GABAergic, so potentially protective.
I have a question about the transplantation of the cells. We know that they have some theory about the GABAergic cells, they keep the circuitry, but what we have like losing the cell death is the MOS cells. So have you checked if some of the cells became MOS cells after they start epilepticus, or the most parts became organelle cells or GABAergic cells?
Specifically examined MOSI cells using markers. We basically looked at the granule cell differentiation in the GCL, and then looked at the phenotype of other cells using GABA markers. Yeah, we haven't examined MOSI cells. It's a very complex cell. It'll be interesting to see whether any of these neural stem cells can make MOSI cells.
Can I ask you, Ashok? So you said that about how much are they? 30% of the cells become neurons. They are mostly GABA neurons. So something that puzzles me a little bit is the improvement in memory. Right? How can you explain that more GABA would improve memory, while clinically, if you benzodiazepines do the opposite? Exactly. And the other thing is, what do you think there is a role of those 60% astrocytes that you get there?
Yeah, improved memory can occur through several mechanisms. GABA is also important for memory. So another thing is these animals are having seizures. So when you suppress seizures, probably some improvement in memory occurs because of that. And the second thing is the astrocytes. They're secreting beneficial neurotrophic factors. Like your favorite factor, BDNF, is also there, FGF2, and then GDNF. All of these are important for improving memory. And then not to forget about neurogenesis. So the transplantation actually prevented abnormal neurogenesis, also maintained normal neurogenesis, closer to control levels. So that may be contributing to certain types of memory, at least pattern separation. I mean, we did a novel object recognition test, and now we are also looking at novel place recognition test that's more specific for pattern separation function. Yeah.
So if I can ask a follow-up question. So you sort of have two phenomena. You're rescuing and preventing some of the pathology with the transplant, and then you have the transplanted cells themselves integrating. Which do you think is the most important factor here? Do you think it's actually the transplanted cells, or do you think it's more the rescue of the pathology?
I think both are important, yeah. Just for suppressing seizures, I think adding GABAergic neurons are very important. But for improving cognitive function, I think rescuing the host cells and maintaining normal neurogenesis also very important.
Hi, over here. Maybe it's a silly question, but gene therapy and cellular therapy, they might promote seizure-free patients and recover cognitive impairments, but you believe inserting these new connections might create a new person, a new personality, since there's new connection, a different memory creation, and maybe a different look to the world.
Want to take that?
Well, the kind of gene therapy that I've been talking about is essentially based on the idea of supplementing some kind of soluble factor, changing the microenvironment in a specific brain area. So, alterations in behavior and personality are obviously possible, but of course they are possible with any drugs acting on the CNS, and actually that may be eventually more likely than with this kind of a therapy that is confined in the distinct brain area, while drugs work essentially everywhere. But in principle, of course, we cannot exclude any possibility.
You want some changes.
You want some changes.
Well, that's a very difficult question, you know. I mean, we do not know at this point, but we are trying to, you know, correct the circuitry in the hippocampus by doing cell therapy. I do not know whether that's going to have, you know, induce a different personality. Yeah.
Can I answer on that? First of all, there are no stupid questions. There are only stupid answers. But I think that was a great answer. but you know, I want to take this opportunity just to expand on how we conceptualize the impact of epileptic treatments and or treatments for epilepsy and changes in personality. If you look at the effect of anti-epileptic drugs on psychiatric adverse events, most of the time it happens in people with a prior psychiatric history or a family psychiatric history. So it's not the drug causing necessarily a psychiatric adverse event de novo. It happens in people who have a predisposition for the development of psychiatric adverse events. Whether a gene therapy can modify the expression of, you know, of genes that have the predisposition to bring up to the surface a mood disorder that was already there, you know, that's a different question. But I think that when we talk about psychiatric adverse events, it mainly occurs in people who have a predisposition.
Yeah, ask a question to Steve, by the way, on this, because you have these P10 knockout mice that have spontaneous seizures and have these alterations in the granule cells that mimic the one that we have in epileptogenesis. And you showed some data on disc 1 knockout with the same phenotype. And so did you check for spontaneous seizures in disc 1 knockouts and vice versa? Is there any schizophrenic tract? I don't know how you can explore that in a mouse, but in P10 knockouts?
So the disc 1 work is in ours, so other labs have done that. And those animals have been studied for a psychological phenotype, and they have, I mean, as much as you can do in a rodent, features sort of consistent with behavioral manifestations. I mean, they have some behavioral issues. I don't think anybody's looked at seizures in the animals with the disc 1. With the reeler, which I showed as well, they don't have spontaneous seizures, but if you challenge them, it looks like they have a little bit lower seizure threshold.
But I was connecting to the comment Andy made. Do you think that your idea is that alterations in newborn granule cells is sufficient to induce an epileptic phenotype, a seizure phenotype, let's say? Do you think that could be sufficient also to produce a psychiatric phenotype?
So, I mean, I'm suggesting that maybe it could contribute to some aspects of some of these diseases. So I wouldn't go so far out on a limb to say that's going to account for everything or even most aspects of different diseases. But what really strikes me is, I mean, given how profound the anatomy of some of these brains is disrupted, it's hard to imagine that that hippocampal circuit is functioning normally. You think that's going to have some consequence. And so I think a lot of more work needs to be done. One of the things we don't know yet is how much these different types of pathologies are characteristic of different diseases, schizophrenia and bipolar disorder and so forth. There's a small number of papers in the literature. So there's, I think, one paper showing an increase in mossy fiber sprouting in bipolar disorder. It hasn't been replicated yet. There's a paper showing an increase in the number of granule cells with basal dendrites in schizophrenia. Also hasn't been replicated yet. So those of you working on human tissue, if you have it and want to look, that would be great to get some more data to see whether that's a real phenomena. So I think that's sort of a key aspect to see, is this pathology even there in these diseases? And then if it is there, that's consistent to maybe to continue to work that out, that disruption of these cells is perhaps contributing to some aspects of these different disorders. Given, or that we know that we talked about earlier, how ventral hippocampus is important for some of the behavioral functions. So, I mean, morphologically, ventral hippocampus is disrupted. So the question is, what effect is that having on the behavior of the animal?
The question I had now was, and this is as much for Steve as for anybody else. We know that being a big believer in circuits and circuit integration, and knowing that after an injury such as status epilepticus, you get a whole denuding of dendrites as the axons die back. And one of the things that we're always considering, there are two pieces of this, is that you get different kinds of axonal reinnervations, and also there's a whole new wholesale change in expression of different receptors and channels that you get changed from NAV 1.1 to NAV 1.3. The sodium channels and the physiology and pharmacology are entirely different. So one of the questions is using kind of the techniques that you all have been working on, which are very powerful, would it be worthwhile to begin looking at not just at the aberrant granule cells, but to begin to look at other areas to see how these axons are re-guided, how you might influence receptor re-expression, because it may be that could be where the key is, that following an injury, that you really want to influence those steps as opposed to trying to put something new in.
So on your final question, I would certainly agree that you would want to look at these things. We start from morphology, so I think it's an easy starting place to try and figure out what the neurons are doing. I've always said that if you know what a neuron looks like, you have a good idea of what it's doing, you know who it's talking to some extent. But obviously, and which we've started to do, to try and do some physiology experiments on some of these cells, and there's a whole host of things, including the channels you mentioned, that could be looked at that may be very important and quite possibly more important than some of just the gross anatomical differences that we see. So to make a comment on one of your comments, that with a lot of the approaches that have been used in the past, you do a treatment and then you compare your treated animals with your untreated animals and infer based on the effect whether your treatment was useful or not. With some of the technologies available now, like the long-term seizure monitoring, it's technologically possible to monitor animals for long periods of time and figure out what their seizure frequency is like, so you can control for these variabilities that you see in a lot of these models. So you're absolutely right that we see quite a bit of variability in the pilocarpine model. So in the recent study we did, we had animals that their status epilepticus was visually identical. We didn't quantify it in any way, but we had animals that ranged from 1 seizure every 10 days to 3 seizures a day. And just looking at the status epilepticus, you'd have no way, I mean, we couldn't predict which animal was going to go which way. But with long-term monitoring, you can kind of figure out what your animal's like after it develops epilepsy, and then you could do various interventions knowing that you have an animal that has a lot of seizures or very few seizures. And I think that makes those experiments more rigorous and powerful.
And I agree. I think if you can do a before and after study, then a careful selection of your animals, making sure you're really being, but for the early interventions, say it right after status epilepticus, that's really not possible, at least not right now.
With that, for early intervention, the variability is an issue. But when we do transplantation in chronically epileptic animals, we select animals based on the frequency of seizures. And we select animals having similar seizures for transplantation as well as sham transplantation. So we have some interesting results in that model too. I did not present that today. Yeah.
But if I can add something on your initial comment, there was that paper in Nature Neuroscience that showed that all neuroscience studies are largely underpowered. And most of our studies, behavior. We use numbers that are, most of the cases, between 10 and 20 animals. And there is an estimate that it should be 100, 150, some really huge and unbearable numbers. So we have been discussing that a lot together, and I believe we should find ways, we believe we should find ways of introducing the rigor of clinical trials in preclinical research. Multi-center, rigorously double-blind, with clear inclusion and exclusion criteria. And on top of that, we probably need models that are more reliable, because, for example, I couldn't agree the more that pilocarpine is extremely variable.
I totally agree with that, yeah. So.
Can I ask what the mortality rates are with the pilocarpine model? I mean, just generally what people's experiences are?
About 50%.
30%.
50.
50%?
No, it's a little less for us. It's about 30%.
Is that one of the reasons why the studies are underpowered? Because you have to do lots of animals then?
Well, I guess on the power, so I think to sort of add to your point is that, I mean, if you just pick a number out of the air or a number of animals that's convenient to do, I mean, it's hard to do studies with 100 animals, but you should, at a minimum, do a power analysis that if you get a negative result, to understand how big of an effect you could actually find, so that if you're going to go out and present negative data, that you sort of know what your experiment can do, so that the more power your experiment has, the smaller an effect you can find. So kind of how well powered the experiment is depends on what size of an effect you wanted to be able to see, but I think there's, I mean, it's something that needs to be done more, is just kind of be aware of that, and so that you can interpret your data correctly.
But did I understand correctly if you were asking if the reason pilocarpine experiments are underpowered is because of the high mortality rate?
No, we just do more animals if we have to.
Yeah. But
the problem is video EEG monitoring. That's
slow,
labor expensive.
Yeah. I mean,
so on the mortality issue with pilocarpine, if you talk to different labs, you'll get slightly different numbers. Rats are better than mice. Mice die a lot. It varies by genetic background. It varies by lab. I mean, it varies from week to week. So a lot of labs, I mean, you can have, and we don't have good explanations for this. There's some literature trying to understand why it might be so variable, and I don't know that we have a clear answer yet. So you can take presumably equivalent animals from your supplier or whatever, and you do the experiment 1 week, and most of them live, and you get 90% survival, and you do the same experiment the next week, and they all die.
Okay, we have time for one last question.
I'd like to make a question to Dr. Kenner, but before, I believe I have an explanation, or at least a suggestion of explanation for this variability observed in the pilocarpine model, is that most of the labs, what they do, they inject the animals, and they just look for the external behavior. They don't record electroactivity, so basically they don't know exactly how long is the status epilepticus. So it's very hard for me to believe just looking at the animal's behavior, because I can record the animals and I see that after diazepam or tiopental, you're going to see the animal still seizing. So basically, if you just look for the behavior, you're not sure how long the animal is seizing. So you have a very large variability, and you have a large rate of death. So it's very, very important to have this paired with the video recordings of behavior. You also have the electrophysiological recordings. It's not easy. There's a lot. I mean, if you want to do number of animals, a reasonable number of animals to do a statistics, you gotta actually set that up in a good way. So I believe that's one of the reasons. Besides the genetic backgrounds and the level of isogenicity that you have in your rats, if your lark is actually doing a good job in terms of keeping the next generation with a similar pool of genetic background from the parents, that you really don't get that. What you see a lot in mice, the background acting on different levels, making more susceptibility of one strain to the other. So I think it's one important thing to record the electrophysiology after injecting pilocarpine. And the question is to Dr. Kenner, is how frequent it is to see psychiatric symptoms in frontal lobe patients, frontal lobe epileptic patients?
So psychiatric symptoms in frontal lobe epilepsy is frequent. For a long time we've been concentrated on the temporal lobe. That's because that's the type of epilepsy that has been studied in a systematic manner in epilepsy centers. 75% of the surgeries are done in the temporal lobe. But the prevalence of frontal lobe psychiatrically related comorbidities is as high as that seen in temporal lobe epilepsy. And depending on which paper you read, some papers would suggest that you're more likely to see problems with attention span, impulsive behavior, poor frustration tolerance, aggressive behavior in frontal lobes, and more problems with depression in temporal lobe epilepsy. But the new studies that have been coming out don't support that. People with frontal lobe epilepsy have as high an incidence of depression and anxiety. And I think the reason for that is because just like our conceptualization of the epileptogenic zone is shifting from one discrete area to a circuit, the same thing is happening with our conceptualization of psychopathology, where in depression and anxiety, you're basically looking at a circuit that involves hippocampus, amygdala, cingulate gyrus, orbitofrontal region, and so far and so on. And if you think from an embryological standpoint, the orbitofrontal region is comes out of the area where the hippocampus comes, so it's the same embryological structure, you see. So I think it's just a matter of looking at phenomena carefully and systematically, and you're going to identify, the same prevalence of depression and anxiety in frontal lobe epilepsy.
So if I could address the model question. So I think that's a reasonable hypothesis that some of the variability that you see in the pilocarpine model reflects variability in the severity of the insult. I think most people use behavioral measures to monitor the status, and I think it's fairly easy to tell when the status epilepticus begins. I think it can be somewhat difficult to tell when it actually stops. So we've done EEG studies in some of our animals, and we've found that even after you treat the animals with diazepam, you may switch a convulsive status epilepticus to a non-convulsive status epilepticus. So the animal can appear asleep, but you look at the EEG and it's still firing away. And we've looked at some of our animals a couple days out after the drugs have presumably worn off, and they can continue in spike and wave activity for at least a couple days after the insult. So there's some tools out there for trying to, do EEG recordings and try and assess how severe the status was. I don't have great confidence in how well those work. I don't know that anybody, it's sort of a problem with the model. There's not really a good way to control or determine how severe the insult actually was.
Yeah, I agree. And what we do, like many other labs, is we repeat diazepam administration after half an hour in case status does not stop. But it is an interruption of convulsive status. And what also we observe is that animals continue having brief spontaneous seizures or even convulsive seizures for those couple of days that you were talking about before entering what we can really call latency. And of course there is a variability animal to animal in those parameters that increase the overall variability.
Yeah, thank you. So with that, we are closing this discussion session. Thank you all for very interesting and insightful comments and suggestions, questions. And I also take this opportunity to thank Norberto and his crew for organizing this fantastic conference. Over to Dr. Norberto.